Depth 109 or 28 meters. At bottom.
At the deepest point on Earth, where pressure can crush steel, scientists lowered their cameras and sampling tools into total darkness. What they saw challenged existing ideas about how the deep ocean works. Life was there, harmful objects were found, and human traces had reached a place once thought untouched. How did such waste reach nearly 11 kilometers down? What survives in that cold, silent water? The deep discovery. The deepest part of the ocean lies inside Challenger Deep in the Mariana Trench. Scientists from several countries have gone down there with manned submersibles and remote vehicles to take photos and collect samples. The Japan Agency for Marine Earth Science and Technology created a deep-sea debris database that holds images and videos from thousands of dives. Scientists use that collection to document plastic and other human items at extreme depths. One clear entry shows a plastic bag photographed at 11,000 meters. Pictures and details in the database list many pieces of man-made waste. Those images come from more than 30 years of diving. The database lets researchers trace how repeated dives have recorded trash at great depths over time. In 2019, explorer Victor Vescovo reached Challenger Deep during the 5 Deeps expedition with his company, Caladan Oceanic. Video from his diving recorded small wrappers and pieces of plastic on the seafloor. That mission also mapped parts of the trench and brought back sediment and water samples for laboratory study. Scientists who reviewed the footage shared how harmful objects might be affecting the lives of animals living in water. China's manned vehicle Fenduje began systematic dives that included the Mariana region after 2020. Scientists from the Chinese Academy of Sciences and the Institute of Deep Sea Science and Engineering collected more than 2,000 samples of water, sediment, and organisms during those missions. Their work produced pictures taken on the trench floor, and samples returned to onshore laboratories for testing. The combined evidence comes from images, video, and lab data. Photos show everyday objects on a seabed that sits under enormous pressure. Physical samples and chemical tests detect tiny plastic fibers mixed in the mud. The two kinds of records work together. Visual records make the problem visible. Lab numbers measure how much material is present. How can material reach such deep places? Several routes move plastic downward. Pieces that float can sink once they collect algae or organic material. Rivers and storms push large amounts of waste into the ocean, and strong currents carry debris far from shore. Undersea flows called turbidity currents will sweep particles down continental slopes. Those flows can push material into trench basins where it comes to rest. Scientists also measure microplastic particles in the water column above trenches. They filter seawater at different depths and detect fibers and fragments far below the surface. Sediment cores pulled from the bottom show layers with tiny plastic pieces mixed among mud and natural particles. Laboratory tests identify polymer types and sometimes link fibers to clothing or fishing gear. The public display of these images has changed how people see the deep sea. The Japan Agency for Marine Earth Science and Technology database made many photos available. Newspapers and magazines published striking shots of wrappers and bags on the ocean floor. That attention led to more fieldwork and more careful laboratory study. Different groups used different tools. The 5 Deeps expedition operated in 2019 with a solo submersible carrying 1 pilot and cameras. The Chinese missions used piloted and remote vehicles with teams that could collect samples on repeated dives. Unmanned rovers and sediment grabs also collected material for laboratory analysis. Pressure-retaining samplers arrived in some research programs to keep microbes and chemicals closer to their natural state during retrieval. Those samplers help when scientists look for tiny particles and for living microbes that might cling to plastic surfaces. Researchers run DNA sequencing to see which microbes live in deep mud and on debris. They use microscopes and chemical assays to classify particles and test for contaminants that stick to plastic. The records include dates, dive numbers, depths, and pictures that allow cross-checking. Research teams compare camera footage with lab results to build a fuller picture. That combined approach strengthens the case that human material reaches the Hadal zone and interacts with local ecosystems. What does the mapping show so far? Plastic has been recorded in multiple trench locations and at different times. Photos and samples confirm the presence of macroplastic items and microplastic particles. The evidence is not limited to a single expedition or tool. It spans decades, teams, and survey methods. Public databases and shared sample sets also let other researchers verify observations. Researchers can request images and lab data, repeat tests, and agree on measurements before drawing broader conclusions. That openness increases confidence in the records. The photo records include entries from depths near 10,900 meters to about 10,975 meters in Challenger Deep. The Japan Agency for Marine Earth Science and Technology Collection contains images from more than 5,000 dives, which helps researchers see patterns over time. Scientists use strict handling rules to avoid contaminating samples with fibers from shipboard clothing or equipment. Scientists bring blank samples and lab controls to separate real particles from accidental contamination during processing. That builds trust. The sight of a shopping bag on the deep sea floor is simple but powerful. It connects life on land to the deepest parts of the ocean. Scientists keep mapping where material collects, and labs keep measuring tiny particles in sediments and water. The combined work gives a clearer view of how human waste moves through the ocean and where it ends up. But if human waste reaches the deepest trenches, what unseen consequences are shaping the fragile creatures already surviving there below? The creatures down there. Life near the bottom of the Mariana Trench looks strange to most people. The Mariana snailfish, Pseudoliparus suirae, is one of the best-known residents. Scientists led by Mackenzie Geringer and Thomas Linley described this small fish after collecting specimens from deep trenches. The snailfish has a fragile body, thin bones, and pale skin. Those traits help it live where pressure is very high and food is scarce. Explorers captured more animals on camera. Victor Vescovo recorded small fish, shrimp-like crustaceans, and worms during dives for the 5 Deeps project. The manned submersible Fenduje and scientists from the Chinese Academy of Sciences collected water, mud, and animals for study. Remote cameras showed brittle stars and slow-moving sea cucumbers on muddy plains. Each clear photo gives a name to a species that taxonomists can study. Microbes make up most of the living biomass at those depths. Sediment samples returned to shore reveal thousands of microbial types. Researchers study their DNA to map genes that help them cope with cold and pressure. Some microbes live on chemical energy that comes from the seafloor. Where methane or hydrogen sulfide rises through sediment, tube worms and bivalves gather. Those animals host bacteria that turn chemicals into food. Where does food come from so far under the surface? Much of it falls from the upper ocean. Dead plants, algae, and animal parts sink as a slow rain. Microbes and small crustaceans feed on this material during its fall. Scavenging amphipods and hermit crabs pick at carcasses on the seafloor. When a larger animal sinks, many species arrive to feed. Cameras and baited traps often record these feeding events. Adaptations are visible in body shape and chemistry. Deep snailfish lack heavy, calcified bones. Their cells contain molecules that keep proteins working under pressure. Several crustaceans have flexible exoskeletons and long limbs to move through soft mud. Many animals grow slowly and produce fewer, larger eggs suited for rare reproduction events. Sampling uses a range of tools. Midwater trawls catch swimming animals above the floor. Baited traps lure scavengers for photography and capture. Sediment cores and grabs collect mud that holds tiny animals and microbes. Pressure-retaining containers help fragile life survive the trip to the surface. Onboard microscopes, quick freeze stations, and chemical kits let researchers preserve DNA and tissue for later analysis. Taxonomy matters because it creates a lasting record. Scientists give new species formal names, record where specimens came from, and publish descriptions that include size, depth, and location. Papers list specimen numbers and authors so other laboratories can check identifications. That work builds a global catalog of life adapted to extreme deep-sea conditions. So far, catalogs include multiple snailfish, diverse amphipods, sea cucumbers, brittle stars, and many worm species. Local hotspots around vents and seeps host concentrated life. Where the seafloor releases methane or hydrogen sulfide, tube worms use bacterial partners to gain organic matter. Clams with symbionts also live near such vents. Researchers mapped these chemosynthetic communities in the Northwest Pacific trenches, listing species that rely on chemical energy rather than sunlight. Genomes reveal how cells survive. DNA sequencing shows pathways that prevent proteins from failing under pressure. Large DNA studies from samples collected by the Chinese Academy of Sciences and other institutions contain many novel genes. Scientists flag certain gene families for closer study to learn which proteins keep cells functional in cold, high-pressure environments. Observations record behavior as well. Amphipods show active feeding around bait. Snailfish hover above the mud, feeding on small crustaceans. Some animals move slowly, an energy strategy in a place where food can be rare. Videos from manned dives and remotely operated vehicles give behavioral ecologists material to study how species interact and how they respond to new food or disturbance. Collaboration speeds discovery. International groups share images, DNA data, and specimens through open databases. Researchers at universities and institutions such as the Chinese Academy of Sciences, the Japan Agency for Marine Earth Science and Technology, and independent groups like Caladan Oceanic publish results that list who collected what, where, and when. That openness helps museums and laboratories confirm names and add specimens to collections. Researchers face hard choices when bringing samples up. If pressure drops, fragile animals and microbes may die. Crews use pressure containers and fast freezing to keep samples useful. In laboratories, technicians run DNA tests, check samples under microscopes, and match specimens to museum collections. That work can take months, but yields clear and reasonable records, including a deeper study. But what happens when the same tools meant to catalog life uncover evidence of human waste infiltrating the deepest oceans? The plastic problem. Plastic reaches the ocean surface easily. Strong winds, rivers, and careless dumping send bottles, bags, and nets into coastal waters. Sunlight and waves slowly break larger items into smaller pieces over time. These fragments move through the water column and can travel far from where they started. Researchers collect seawater at fixed depths and pass samples through fine filters to count particles. Crews also recover sediment cores that capture layers of mud and trapped material from the seafloor. Those cores often contain tiny plastic fibers and small fragments mixed into the sediment. How do heavy pieces reach the bottom of the deepest trenches? One reason is biofouling. Tiny organisms attach to floating plastic and add weight until the item sinks. Another reason is aggregation. Plastic sticks to dead plankton, algae, and marine snow and falls faster as a clump. Storms and floods push large amounts of waste from land into the sea, sending material into offshore currents. Powerful undersea flows carry debris down continental slopes into deep basins. These sediment flows, called turbidity currents, move particles long distances and deposit them in trenches. Scientists use clear methods to find and classify plastic at depth. Research vessels collect water with sampling bottles and pass the water through fine mesh filters. For the seafloor, researchers use corers that pull a column of sediment without mixing layers. Onboard technicians sort particles under microscopes to record shape and size. Chemical instruments, such as Fourier transform infrared spectroscopy, identify polymer types. That testing shows whether a fragment is polyethylene, polypropylene, polyester, or another common material. Microplastics cause particular concern because they move easily through food webs. These pieces measure less than 5 millimeters and include fibers from clothing and ropes. Sediment cores from deep sites show microfibers mixed into mud layers. Plastic content can vary widely between nearby spots and with depth. Scientists report counts by number, by volume, or by weight, so that remote sites can be compared with coastal places. Methods differ between laboratories, so researchers use blank controls and clean room procedures to limit contamination from clothing and shipping equipment. Plastic also carries chemicals on its surface. Manufacturers add plasticizers and stabilizers that may remain attached to fragments. Metals and organic pollutants can adsorb to plastic and travel with particles. When animals ingest plastic, those chemicals can enter tissues and move through food chains. Filter feeders and small scavengers that eat sediment may take in microplastics with organic matter. Researchers examine stomach contents of deep crustaceans and fish to measure ingestion and then run chemical tests on tissues to check for transferred pollutants. Scientists call colonized plastic the plastisphere. Microbes settle on pieces quickly and form communities that differ from those in the surrounding mud. Bacteria, fungi, and tiny single-cell organisms live on plastic surfaces and create distinct groups. Genetic analysis of DNA from plastic pieces shows genes related to breaking down complex molecules. Researchers search for those genes for enzymes that can attack plastic polymers. A few enzyme families, including PET hydrolyzing proteins, show activity in laboratory tests using marine microbes. Sediment layers act as a record of pollution over time. Radiometric dating and other techniques let researchers match buried layers to years or decades. Combining footage from manned submersibles and remotely operated vehicles with sediment chemistry gives a fuller picture of when debris arrived and where it concentrates. Work by the Japan Agency for Marine Earth Science and Technology, the Chinese Academy of Sciences, and independent groups like Caladan Oceanic links human waste to the deepest seafloor. Observations range from whole items resting on the seafloor to microscopic fibers trapped in mud. Sampling, chemical identification, and DNA testing provide the details scientists need to map pollution and study how these materials directly affect deep life and sediments. But when scientists lift DNA and images from abyssal depths, how does discovery turn into obligation, policy, funding, and accountability? Real-world impact. What happens when science brings up living samples, DNA sequences, and clear photos from the deepest ocean? The work shifts from curiosity to projects with rules, budgets, and goals. Laboratories at universities and at King Abdullah University of Science and Technology screen deep-sea genetic data for enzymes that can break down common plastics. Those proteins operate under cold temperatures and high pressure in their native setting. Scientists test whether related proteins can be adapted for use on land. Who holds responsibility to act? Governments, research institutions, and ocean agencies must address pollution that reaches extreme depth. National funders, such as the United States National Science Foundation, support projects that map plastic pathways and measure impacts on deep life. The Japan Agency for Marine Earth Science and Technology makes image collections available so independent researchers can verify records. The Chinese Academy of Sciences supplied thousands of water, sediment, and organism samples for laboratory work and genetic study. How could enzymes from deep microbes help industry? Microbial proteins from cold, high-pressure habitats may show stability that standard enzymes lack. Protein engineers test such proteins in controlled settings to see if they speed the breakdown of polymers, like polyethylene terephthalate. Early laboratory tests using related enzymes show slow activity at low temperatures. Researchers then change sequences to raise activity, a step that needs careful safety checks and long trials. Any applied use must pass rigorous testing for safety and efficiency before it reaches recycling plants. Policy questions follow scientific work. Evidence of human material at trench floors talks about ocean protection and seabed use. Countries and international bodies consider rules for deep-sea mining and for sampling practices. Conservation groups and resource planners now include deep trenches in risk assessments. Some policymakers press for stricter controls on waste release at sea and tighter rules on single-use plastics. Regulators must balance scientific results, industry needs, and social concerns while drafting effective measures. Pollution studies themselves improve over time. Researchers build better samplers and controls. Pressure-retaining containers offer more reliable microbial samples. Clean room protocols reduce contamination from clothing and ship equipment. Standard laboratory methods help compare results from Japan, China, Europe, and the United States. That consistency strengthens global surveys and helps prioritize where to send ships for focused work. Economic and ethical issues appear with the search for useful enzymes. Who owns genetic resources collected in international waters? Draft rules and treaties aim to balance scientific access with fair sharing of benefits. Institutions that supply samples often partner with laboratories and sometimes with private companies. Benefit sharing can include joint research projects, training, or shared revenues from any commercial products. Clear agreements and open contracts reduce disputes and make collaborations smoother. Practical next steps are simple to state. Scientists will map plastic movement, test how sediments trap particles, and screen deep genomes for useful proteins. Regulators will review waste rules and seabed policies. Funding agencies will support long-term monitoring cruises. Private laboratories may partner to adapt marine enzymes for recycling plants, while universities continue basic studies on how deep life copes with pressure and scarce food. Clear sample handling and open data sharing will matter. Museums and national laboratories will archive specimens so researchers worldwide can check results. Collaboration across countries will speed reliable answers and reduce duplication of effort. Many institutions are involved, and many steps lie ahead before any laboratory result becomes a product or a regulation becomes a global rule. From the deepest trench to daily life on land, this story shows how closely we are tied to the ocean, even when it feels far away. What ends up miles below the surface starts with choices made above it. What part surprised you most? Drop your thoughts in the comments. If this helped you see the ocean differently, like and subscribe for more. There is a lot more below the surface worth talking about.